my name is Kamora Minor. Currently on my I don't even know take of this video. I'm from section 007 and I have the protein of KRAS with the mutation of replacing the glycine at number 12, position 12, with either cytosine or aspartic acid. Usually the KRAS protein is responsible for signaling for cell growth, which is important for cell survival and cell division. While it is seen that by replacing glycine in this particular position, the cell becomes less flexible and is constantly in its active position, causing uncontrollable cell growth known as tumors or cancers, because some tumors are cancerous. Now, KRAS is known as an enzyme. We all know that enzymes catalyze reactions. This enzyme catalyzed the reaction from GTP to GDP, making an enzyme. It is usually located in the inner plasma membrane of the cell, usually of epithelial cells, in organisms considered to be animals, and this is a part of the cell signaling system. There's other things under that umbrella, but it's usually part of the cell signaling system. Sometimes the immune system or growth regulation. As I said before, with this mutation, the KRAS protein stays in the active GTP bound state. Usually it switches between active and inactive, but these mutations prevent it from turning off, leading to uncontrolled cell division and things. With these mutations, the protein itself becomes bigger, less flexible, and unable, as I said, to fit in small space. It is seen that the two amino acids that replace glycine in this particular mutation are both bigger in size than glycine, but they're like in medium compared to the other amino acids, but they are both bigger than glycine, which is why it becomes like a bigger cell. Usually, it is seen that this protein only participates in like peptide bonds or weak van der Waals interactions. But with G12D mutation, which is aspartic acid replacing the glycine in position 12, this introduces a negative charge which alters the structure altogether. It also introduces a carboxyl group rather than the hydrogen group that's usually with glycine. With the G12C mutation, glycine is replaced by cytosine, which can form disulfide bonds, which further affects the protein structure. It is seen that both mutations prevent KRAS from returning to its inactive state, as said before. The difference: the normal KRAS protein is small, neutral, and nonpolar, while both G12D and G12C become polar and hydrophilic. But G12D is negatively charged, while G12C is neutrally charged. They both become less flexible. They're both bigger. They both can't fit into smaller spaces. It's just the smaller differences. We're going to go to the model now. This is the model. I chose to compare, instead of doing one, I chose to do the comparing of the original versus the mutated. This isn't a specific one. It's not G12D or G12C. It's just solidifying the differences between the mutation versus the regular. So this is a regular. It has like single signaling. It's small. It's flexible. While this one, it's bigger. It has multiple cell signaling, as we know before, because both these mutations stop the protein from going into its inactive state. This causes like the growth. We see it's bulkier. We see it's less flexible. We see all of this due to the side-by-side comparison. Let me back it up. I chose the colors to represent like the multiple signalings, and I chose this one as like the normal. You can't really see, so the limitations of this are a lot. One of the biggest ones is you can't see like the specific cell interactions. This, I if I knew how to, I would have. And if I would have, it would have solidified the effect the mutation has on the function of this protein. But I didn't know how to do that. Quite frankly, originally I didn't even know how to make this protein a thing in 3D form, but I tried. This also doesn't tell the whole story, I would say, of the major differences, but it does like point out the biggest ones that show like a big difference, and it explains as to why kind of it this stays in the active form. Let's put that down. Let's do an overview so I can make sure that I said everything that I needed to say. The KRAS protein usually is a cell regulator. It controls the cell signaling of cell growth, but with mutation of G12C or G12D, which is replacing glycine in the position of number 12, particularly, the protein is unable to go into its inactive form, causing uncontrollable growth, which can either be cancers or tumors. It's usually in the inner plasma membrane of epithelial cells of organisms that are considered animals, also humans. It is also a part of the cell signaling system, which has other things up under that umbrella, but cell signaling system. KRAS protein is considered an enzyme because it catalyzes the reaction of GTP to GDP. Usually the glycine of this reaction has a hydrogen as its bonded group, but with G12C, which is cytosine, that has a thiol group, and G12D, which is aspartic acid, has a carboxyl group. Both G12D and G12C are bigger in size compared to glycine, but they aren't the biggest of the amino acids. They're more in the middle, but they're both bigger. This mutation causes the protein to be less flexible, bigger, always active. Compared to glycine, G12C is able to create hydrogen and disulfide bonds, while G12D could create hydrogen bonds. But compared to its originality, the KRAS protein, the glycine could only create peptide bonds or participate in weak van der Waals interactions. I do believe that's it. This idea about the mutation of this protein is very important to know and understand so that we could move forward. We can make plans to see how to treat this type of growth, cancer, find the root of these problems, and help people in general.